we've listened to several talks with lots of information, so let's all take a deep breath, including myself. I had the privilege of being up here earlier today with two people who I look up to in Yale Weiss and Alison Berendt. They're amazing advocates and parents and pioneers in bringing forward medicines for patients with rare disease, and I obviously can only hope that I can follow in their footsteps with pushing forward a therapy for Dup15q syndrome, and that's what I want to talk to you about today. And I'll be in the hallway wandering around after this. If you have questions, please come up and ask. I wore more casual clothes, so I look more approachable. So please, I'm very happy to talk and answer questions. And I've been doing this with the Alliance for quite a long time, and actually a lot of credit to Alliance for giving me the opportunity over the years, including now, to be able to stand up here and speak to you all. So you are your brain. It sits in your skull. There's lots of parts to your brain. There's many different cells that make up your brain. Have you all heard of cells? Great. That's the level which we need to know for this talk, just so you know, as a warning. But of course, the way that your cells communicate is through electricity. So please raise your hand if you or one of your loved ones or your Dup15q family member has had an EEG. Great. So a lot of people here are very familiar with sort of the gross output of this electrical activity. EEG is something that goes on the skull to measure the electrical activity because we cannot actually access the brain below the skull. Until around 2006 and 2007, where Nobel Prize winning work discovered that you can take blood and skin and turn that into stem cells, and then those stem cells can be turned into neurons, which are brain cells. And that means we can actually take blood and skin cells from those living with Dup15q, where we otherwise wouldn't be able to study the brain unless we are using an EEG, and we can turn those into neurons or brain cells that we can then study. And this is exactly what we do. And so I actually also, if Combined Brain is still around, urge you to go volunteer to give blood. Even if you're a healthy family member, it's often very useful to have a sort of genetically related comparator when we're studying Dup15q in what we call in a dish. But the brain these electrical signals are what allow me to speak to you right now. They're moving my tongue, and they're allowing me to stand, and they're allowing me to maintain my posture and sense my proprioception and where I am in space, and it allows me to think and have memories, and it's allowed me to imagine a future. But these are the things that are altered in disease, ultimately. Your proteins might change and your genes might change, but those things impact how the neuron functions. And so what this means is that your brain is really impacted in all neurological diseases. If you have misfiring in your neurons of the brain that are related to your spinal cord, you can have impaired sensation of pain, whether that's lack of pain, which for those of you that watch Grey's Anatomy have seen the one case of the patient who feels no pain. That's actually, seems like a great idea, but it's actually deadly. And you can have excess pain. So you can live in pain all the time, where touching a feather will induce pain. Imagine living like that. There's epilepsy. This is like excessive firing and electrical activity of neurons. These are the things we're measuring in EEGs. These neurons or brain cells have connections to each other. That's how they speak to each other and talk to different parts of their brain. And those are impacted in autism and neuropsychiatry, so depression and anxiety, et cetera. And then, of course, when brain cells start to die, you get neurodegeneration. This leads to things like ALS, Parkinson's, and Alzheimer's disease. And I mentioned that it's very difficult to study the brain, but we've developed the technology at Quiver Bioscience to do this in a scalable way that makes drug discovery accessible. And our ultimate goal is to start with a potential library of medicines, put them through this magic box that you see here called the Quiver Firefly, and then wind up with a magic molecule on the other side that's a medicine that will go to patients. Obviously, it's not that simple, and we'll talk about the challenges of doing that. But really, our goal is that we believe we can find a more effective and safe and translatable medicine because we're using patient cells to study the patients rather than a cell that has nothing to do with the brain. which researchers often use, by the way, because they're accessible and fast and easy to work with. Now, what's in this black box called Firefly? Well, the process that we use here, again, we start with the patients. Those blood and stem cells get turned into brain cells or neurons. And then we apply drugs to those neurons. And our machine measures these signals labeled in number 4. On the top, these are just all cartoons. So on the top, this is not real data per se. But the top is what a signal might look like from a neurological syndrome, such as Dup15q. And our goal is to treat those cells such that they behave like the purple, where the excitability is maybe not so severe. And this is what the technology looks like. So this is a movie. And every color, blinking color on this screen is an individual cell from a set of blood samples that were turned into neurons. And this technology was never possible before Quiver Bioscience. There's similar technologies, and I'll talk about the Alliance Fellowship, which funded my graduate work, where I painstakingly sat next to a big machine, looks like I was flying a plane, and used a little video, like a video game controller, to move this tiny electrode that's microscopic all the way towards the cell, one at a time, 90 cells a day, for 7 years. And my very first day coming to the company, this is the reason I joined the company, is the very first day I sat there. In one slide from one experiment, I saw 10,000 cells that we captured in just 10 minutes. And that was the entirety of my PhD sitting there in front of me. So this is a powerful tool, because when you can measure cells so quickly with such high resolution, it allows you to screen drugs. Because it's not like I'm going to pick a drug out of a basket and that's the perfect drug for anyone. It's more complicated than that, right? The chances of me going out in the world, taking a drug and saying this is going to work for Dup15q, that's slim. But if I can screen lots of drugs, I can find the ones that are going to work. And then it's up to us to do the requisite studies to find the safe molecules, which I'm going to largely talk about today. So just a note, I am not a family member of an individual living with Dup15q. But I've had the privilege of meeting hundreds of family members and Dup15q individuals over the past decade, and that's because in 2015, I was one of the inaugural Dup15q Alliance Fellowship awardees. This picture was taken in Redondo Beach, because 2 years later I got to present on my work, but the award was awarded in Orlando, Florida in 2015, so 10 years ago from this meeting. And I think that's quite striking, because Shefali said earlier that that was the conference she first came and took measurements and worked with patients on site in Orlando. So we were at the same conference at the same time. Back then, I was scared of her and barely knew her. She is a hero in the field, and I'm now obviously very proud that she is a colleague of mine. So just how does this relate to Quiver? Well, that 3-year, $75,000 fellowship supported building cellular phenotypes. And you might have heard this a lot yesterday in the science panel. A phenotype is just an observed difference between what you're studying and sort of the wild type / control condition. So we're just looking for differences that, in a dish, might represent what's happening in the real brain in the patients. And we use these patient-derived cells. So these came from people out in the world living with Dup15q syndrome. And we observed this difference in the graph. So there's a frequency. Frequency is the number of those electrical signals. And you can see they're quite strikingly different in purple in Dup15q than they are in control. And very different than what you see in Angelman syndrome, which you all have heard about. There's Dr. Eric Levine, my mentor. He was at the science conference, another incredible Dup15q community scientist. And we published this paper in 2021 that it's a very complicated name, but ideally it just means that Dup15q cells they have a lot more of this electrical activity than the wild type cells. And I just want to refer back, if you could notice this really happy, excitable guy in the background here. I used that back then as a way to represent this excitability. So you can imagine just being excitable all the time. It's great to be excitable sometimes, but it certainly don't want to be excitable all the time. Now, I just want to take 1 second to take a nod to the Alliance, because that seed of funding me in 2015 has led now to an entire company that has a lead program in Dup15q. There's 30 of us. I didn't count this as 30, but I asked ChatGPT to make me a 30-person version of the one on the left. But that, there's impact there, and I just want to highlight, please, every little bit of help that people donate to the Alliance, it goes a long way in terms of the impact it has down the line. And certainly this is not one we could have predicted, and I certainly didn't predict myself in 2015. So here's some of the Quiver team. This is an inaugural Boston area Believe Walk that my wife and I hosted in September of 2024, where we raised around $25,000 for the Alliance. And Quiver has been Believe in the Cure Gala sponsor for 2 years in a row, 2024 and 2025. We also have had the privilege of hosting Sarah Thacker in person and Jackie Vanderhoof remotely at our 2024 Rare Disease Day. And why I want to show this is to let you know that this is not just a company of people who don't know what Dup15q is and they're just there to collect their paycheck and go home. People showed up for this walk because they really wanted to meet the families. We listened to Sarah and Jackie talk about Dup15q because we really care about the patient experience. And we support the Alliance ourselves and raise money because we really care about the broader mission of the Dup15q Alliance as we also move forward a therapeutic. So I want to introduce you to some of the humans that are behind the work that's getting done at Quiver. These four incredible people, Aishwarya Dandapani, Karthiani Hari Krishnan, Jennifer Grooms, and Jane Jock, these are the people who work with the Dup15q cells every day in the lab. So these are the people who are doing the experiments, and they are as aware about this community as I am. And I'm their manager, so I can promise you they have to listen to me talk about Dup15q ad nauseum. We also have an amazing team of scientific leadership and guidance at the company, ranging from drug discovery experts to bioinformaticians, cell biology experts, and actually we've managed to recruit a former UB3 expert, Noel Germain, who was a postdoc at UConn when I was a student, and many years later we're now united at the same company. Now, this trust building and working together and therapeutic development companies talking to patients has led to a really important moment, and this is the sort of impetus for this talk, which is earlier this year we announced a partnership/collaboration with the Dup15q Alliance to advance an antisense oligonucleotide therapeutic program for Dup15q. And it's been advancing for quite a while, to be clear, but we'll talk a bit about exactly the type of work that the Alliance has funded to help get this medicine closer to those in this room. So now let's talk some science. UB3A is a gene. Have you all heard of a gene? Okay, great. We're on the same page there. It's increased in Dup15q syndrome. And there are a lot of genes that are increased in Dup15q syndrome, but I think what Allison said this morning is important, is that you have to start somewhere. Scientists love to do experiments. We could do experiments for another decade, but you certainly don't want to wait that long. So starting out with one of the genes that we think is the most likely impactful gene in the area is what we've done. Our goal is to reduce the levels of that gene, UB3A, back down to baseline levels. Since UB3A is a gene, it's composed of a long string of DNA letters. And I mentioned this CSI example earlier today. Just like your DNA is specific to you, it's also specific to a gene. So the UB3A string of letters is very different than the string of letters that makes up UBE3B, which actually is a gene. These letters are A, C, T's, and G's. So now we're going to pause. Have you all heard of the A, C's, T's, and G's? Yes. Okay, great. Well, maybe not all of you, but still a good portion of you might remember this from biology. So that's what the gene's made up of. But that's also how we make up the ASO. So when you hear antisense oligonucleotide, nucleotide just stands for those 4 letters. It's a string of those letters, and oligo meaning many of them, right? Or you have a monomer as a single one, but ASOs are several letters. The more letters you have in the ASO, the more likely it is to be specific to UB3A. It's very easy to find somewhere in the genome that says AAA. But it's much harder to find something in the genome if it's 20 letters in a row that are exactly identical to the area of UB3A we're targeting. And this is important because we want to reduce UB3A, not all the other genes in the body. Those other genes, I mean, there's some other ones in the duplicated region, but there's it would be like 18,000 or 17,850 or something genes that aren't changed, and we don't want to touch those. So ASO stand for antisense A, oligonucleotide, and they're anti to the target, in this case UB3A, and thus we can specifically look at UB3A, and then they should be able to bind and trigger degradation. So that's what this looks like. This is a ChatGPT generated cartoon, again. So you see a series of letters. Those are not real letters on UB3A, but you understand the point. And then there's this other complementary piece that's called the Gapmer ASO that has complementary letters to A, C, C, T, G, G, et cetera. You've heard of, maybe you've heard of Watson and Crick. They've discovered this complementary pairing, and that's what this is. It's complementary to the DNA. And then this thing called RNase H at the bottom is something that lives in your cells, and it degrades these types of genes, and then they no longer are in the cell. So what that means is if we bind enough ASO in the cell, this RNase H will come along and it will reduce UB3A. So here is a cartoon. This is not real data, but this is just to explain. So on the left in purple are control and baseline levels. And next is Angelman syndrome, which results from deletion of an area that houses UB3A. And so UB3A should not be there in this cartoon. And then there's two flavors of Dup15q, which all of you are familiar with. Interstitial, in which there's one extra copy on the maternal allele. So now there's 2x expression of UB3A. And then isodicentric, which is 3x. And I know there's a lot of other variants, and I learned about that at this conference talking to families. But this is the sort of basics, let's call it genetics 101. And our goal is to put an ASO on cells or give an ASO intrathecally to patients to reduce that UB3A as best we can back down to the baseline levels. And I think just between the difference of general phenotypes across isodicentric and interstitial, we know that there should be therapeutic effect even between or before we get exactly to 1x. So I just want to be clear about how much room we have to work with in terms of improvement. It's not like we need exactly this much. Pretty much any improvement we have is going to lead to, I believe, clinical improvement. Of course, we have to prove that, but that's our goal in thinking. So here is real data. The left-hand side are 3 different wild-type neuron samples. So these would come from a family member or a matched control. And then in the increased area are neurons from different Dup15q patients. Each color is a different patient. And you see that, as expected from the previous slide, where the isodicentric are 3x, the real data also says that there's 3x UB3A in these cells. When we put ASO on these cells, there's 2 different ASOs. We can bring UB3A exactly back down to the control levels. Those are on the patient cells. The other important thing is these ASOs are tunable. So it's not one dose and that's all you have. You can give different amounts of ASO and control how much UB3A you can express. So this is really good because, of course, the dose that you might give an individual living with isodicentric is going to be different than that you would give to an individual living with interstitial. These are on cells, right? We're not doing this yet in patients. It's more complicated, of course. But what that shows you is that we've already finished all the designing and screening. Screening meaning we're like looking for all the types of ASOs that are going to do exactly what I just showed you. So that part's done. On the left-hand side is a funnel for reference. The next thing we want to look for is what we call efficacy. And you heard Allison talk about this as making sure the drug does what we want the drug to do. So it's not just enough to show that it reduces UB3A in a cell. You want to show it's correcting behaviors of the cell. Very different than correcting patient, but it's still a proxy for what we expect to happen in a patient. So for instance, the cells do lots of different things. If we don't correct any of that, but still knock down UB3A, it's hard to believe that just knocking down UB3A in a complicated individual like a human is going to actually lead to any therapeutic benefit. So we want to show that we can change the phenotypes in cells. And those are the same phenotypes that we studied in Eric's lab in 2015, supported by the Alliance. So this data you see here on the left-hand side, this comes from the same exact samples that the Alliance funded in 2015. So when I and my team worked with these cells, we were taking vials of cells with my initials from the distant past. And what you're looking at here in the first column, where the colors are a lot darker, are every feature that we measure with that instrument called the Firefly. There's 350 features here. And this is a variety of features associated with how the cell is behaving in a dish. If it's purple, that feature is greater than the wild type. If it's green, it's less than the wild type. If it's darker, the distance is, the magnitude is larger. So the darker purple means things are really lower than the wild type, and the darker green, et cetera. And then the next 4 columns are ASOs 1, 2, 3, and 4, all treated at a certain dose. And what you're looking for and what you see is that all the colors flip. And that's exactly what you want. You want to show that all the features are now reversed from what they were before. And this is important because it's not just one feature that's changing here. It's all 350, or a majority of the 350 features that are changing. And I think this is important because what it means is we haven't touched GABA. We haven't touched HERK2, and these are genes that are in the duplicated region. And yet we're still changing all the features. And what this makes me hopeful for is that an ASO, as Allison was saying, isn't just seizures or isn't just muscle tone, but we're hoping it's going to be disease modifying. Another way to visualize this is in high dimensional space. So all you need to know here is that this other way to visualize this data is the difference between the blue and the orange. And there's 2 axes on which you can be different. There's the y-axis on the left-hand side, and there's the x-axis on the bottom. The yellow, blue, green, and purple dots are the Dup15q cells plus an ASO at 1 dose. This is with 0 optimization. We haven't done any experiments on seeing whether or not we can change the dose or when we give the ASO, et cetera. And just from 1 experiment, we show that, for instance, the yellow ASO has fully brought the Dup15q behavior up to the same y-axis area as the control, and a majority of the way on the x-axis, which is really encouraging. By the way, there are 7 other what we call lead ASOs. So these are 4 candidates, and we're assessing safety of a bunch of them. So we actually are going to be testing the other 7, and we believe maybe they can be even better. But again, there's still timing and dosage that we're improving. And I believe that we'll get full rescue based on this initial data, but stay tuned. So that's efficacy. So we have ASOs that reduce UB3, and we have ASOs that in Dup15q cells that have a variety of behaviors. We can change all of those behaviors in cells with the treatment and reduction of UB3 without having yet optimized the dose and the timing. And now maybe perhaps the most important part, which is minimizing toxicity. So this is the study that was funded by the Dup15q Alliance. And there's a lot of other graphs I can talk about. But you guys, again, have heard a lot of information today. So we can keep it short and sweet. One of the studies that's really important before you can put an ASO into a monkey, and a monkey is a really important study because that's the sort of bridging species before human, is a rat. And the rats are really important because they're big enough that you can give the ASO intrathecally, which is also the way that the ASO would be administered to patients. In mice, they're very small. It's much more difficult to give it intrathecally. We've done other mouse studies. But this is one that's now much more proximal to how the ASO will be delivered to the monkey and eventually, and hopefully, patients. And so there's a paradigm that's been established by companies like Ionis. Now, Ionis is the inventor of Spinraza. I'm going to do a test. Do you all remember what Spinraza is from the last couple days? OK. So Spinraza is the drug that people have described as a miracle drug because it was an ASO for spinal muscular atrophy, where patients often would die quite young and now are living because they have an ASO. It's the sort of bright star we all look towards when we're making the ASO. So the developers of this really special and incredible drug have developed a paradigm to make sure that these types of drugs are safe throughout drug development. And one of them is to give the drug in very, very, very high doses to rats. And these are doses that the monkey will never see and that patients will never see. But the thinking is that if this drug is safe when you give a ton of this drug, it should certainly be safe when you give a lot less of it. And so here is real data. I don't want to just tell you, hey, we did this study. All you need to know is look for the numbers that are green or red. Red's bad. So, and the good news is we don't want all 11 ASOs to be green because then we'd have a hard time. We can't develop 11 drugs. That's going to cost like $500 million. We want 1 drug. So all we need is 3 of these to be green for a couple more weeks. This study is ongoing, but a couple more weeks, and then we would move 2 to 3 of those into a monkey. And from the monkey study, we will declare a single, what's called development candidate, which is be the one we'll have discussions with the FDA to initiate what are called IND-enabling studies, and those are very important. So thank you so much to the Alliance for being supporters of this very critical study, and this is the type of study that gets investors excited as we go out and raise the capital to do the more expensive studies. I'm very encouraged by this. The first group gets sacrificed next week, so I think we should have some drugs on our hands. Okay, so we've minimized toxicity. We have 4 minutes and 39 seconds left. So this is what's left to do. Non-human primate, IT, tox, and then non-human primate IND-enabling studies. So as mentioned by Allison this morning, a lot of time, effort, and money has gone into getting just to this point. I've given a talk each year, progressing more and more through this funnel, but there's been a lot of man hours and a lot of money and time that's gone into getting just to this point. And there's not many steps left before clinical trials and getting this therapy introduced to patients, right? There's 2. However, these steps are some of the longest and most expensive, but they're also the most critical, because you can save rats and mice and cells all you want, but it's not going to matter if an ASO isn't safe in a monkey. I can promise you it will not be approved. So now we're out fundraising a Series A to get this program all the way to the clinic. So that's the work that we're doing right now. So I'll end with a brief reflection and sort of how I think about these things, which is in 2015, when I was still a student, I never had heard of Quiver Bioscience, so that's one thing. I think like 25 or 30 researchers came to the science meeting at that time. The family presence was quite large, but just a few researchers. I think there is probably 1 paper published every 3 or 4 months by a sort of well-recognized, sort of highly impactful scientist. And I had never heard of Spinraza, nor was it approved. And ASO was not something anyone at the Alliance or even my thesis advisors had ever heard of. But here we are, 2025, and not only is Spinraza approved, but there's 3 or 4 other ASOs that have been approved for CNS indications, including ALS. And there are several others that are in late clinical trials that are, sounds from this morning, helping patients walk and swim independently and have words when they previously didn't have words. There are, I think, over 100 people in attendance at the science conference. And there's probably been 100 papers or more published, maybe 1 or 2 per month, on Dup15q sort of since the last several years. So I'd say that a lot has happened in what may seem like a long time, but really, in fact, is quite a short period of time. And now you have several companies in attendance at this conference, including the talk from Sophia just before about this really promising therapy for severe seizures, and 2 potentially disease-modifying programs between Kicho and Quiver that are really close and within reach, where we can now start to get excited as a community about it's far, but it's really in sight, right? And so if there's anything I hope I can leave you with today is that there's a lot of people outside this direct community that are in your corner, and we're working hard for you every day when we go to work. Thank you. Time for questions? It's coffee time. Maybe we can just go get coffee and you ask me questions out there. How does that sound?
Okay, first of all, thank you for doing what you do for our kids. Okay, I think Allison mentioned earlier that, why is the current way to deliver the ASO through a lumbar puncture versus just IV? I know she mentioned that if you do it through a lumbar puncture, 20% gets to the brain versus if you do it through IV, it's more like 60%.
Oh, yeah, great question. So that's exactly what she said. So thank you for clarifying. But what she was referring to is actually a an early technology that we were hearing about in the science meeting. So this is actually not ready for humans. So one day we hope that we can do this IV. So right now, actually, if you today give an ASO IV, and the amount that would get to the brain would be almost 0. So that's why we're doing it intrathecally. And the point that I was trying to make when I brought up that technology earlier this morning is just that the question I think was raised from the left side of the room was like, well, we have to be taking ASOs all the time for the rest of our lives. And I think that's the case for generation 1. And that's the point of Allison. So this is what we have today. We want to get a medicine into patients. We need to start instead of waiting. But the point I'm trying to make is that that technology is only going to get better. There's people who are also trying to make devices that are implantable, that you actually never have to go for an injection, and the device will just deliver ASO. Now, Quiver's not doing that today, but that's the technology that's on the horizon. So I believe whatever regime patients will be on in the next several years for ASO, that's almost guaranteed to change in a better way. So whatever we have now is the worst it ever will be, because it only is going to be better, more potent, and more durable.
One question regarding I've heard a lot of mice production with Dup15. When you are testing in rats or in monkeys later, are there also monkeys that have Dup15q, or that's only to see if they don't have side effects or whatever?
Exactly. So that's a great question. There are no, well, you can make a monkey with Dup15q, but working with monkeys is extremely regulated, right? They're higher order species, it's really hard to find places you can do it, and manipulating them genetically it's really difficult and very costly. Doing studies with like 1 or 2 monkeys costs like millions of dollars sometimes. So certainly making a disease model of will cost a lot of money. So we use the monkeys for safety. You know, they're larger than rats. It's the same, often some of the same anatomy. But the point is that if we've shown through mouse models and other ways, approaches, that UB3A knockdown is rescuing more than one thing, and it's having an effect. Not just lowering UB3A, but having effects. It's very reasonable to believe that in a monkey, even though it doesn't have Dup15q, as long as we're showing it's safe and is still changing UB3A. So not just safety, we'll be collecting tissue from the NHPs and showing that their brain tissue has lower UB3A. Those are sort of the major drivers of that. One thing we can look at, though, because it's not starting with Dup15q, and this is important, is that when we give these ASOs to the NHPs, they're not starting to show Angelman-like effects. And there's a lot, because going too low cannot be good, right? There's a sweet effect here, a Goldilocks sweetheart effect. And I'd say that a lot of the data I've seen and some of the data we've been collecting suggests that when you do this later, rather than so Angelman kids are born without UB3A. They don't even have UB3A in the womb. That's not the case here, right? We'd be giving UB3A to kids that are 1, 2, 3, 4 years old and older, so they've already developed with UB3A present. So we believe that knocking it down, even if we were to knock it down really low, it would not induce Angelman. And second is, it's really hard, as Allison said, to even get enough ASO into the brain to have probably go from 2 to 1, yet alone even further down. But yeah, that's the current state of how the monkeys work.